Hi, everyone. My name is Wynn Meyer. I'm an assistant professor at Lehigh in Eastern Pennsylvania, and I'm excited to talk to you about some of my ongoing work to identify molecular evolutionary signatures of adaptation to different diets in mammals. A major question that motivates my lab is what's the molecular basis of adaptation to dietary specialization across mammals? As you can see here on this cartoon of a subset of the mammalian phylogenetic tree, mammals have adapted repeatedly to a wide array of different diets, with different diets on this cartoon represented by different colors and symbols and spread across the different species of the mammalian tree. So what you can see here is that many diets repeat and have evolved convergently. And this enables us to look at molecular evolutionary patterns across these distinct species to identify genes that are important for that same diet in these different lineages. The tool that we use to do this is called RER Converge, and the pattern that it is able to detect is illustrated in these series of figures. So let's assume that on this tree, representing an individual gene tree, the foreground species of interest are represented in green. So if this gene is important for adaptation to a particular diet, let's say, that is shared among these green branches, then this gene will show accelerated evolution specifically on those branches of the tree. This enables us to compare the evolutionary rate on these foreground branches with the evolutionary rate on the background black branches, correcting for the expectation based on the genome-wide evolutionary rate. So this leads to a test statistic of this particular foreground branch in this for this particular gene with the expectation based on all genes within the genome. So this is the same foreground branch labeled with this arrow in this plot and this plot. And then we can use a test statistic that is essentially a corrected residual of the gene-specific rate of evolution with the genome-wide rate of evolution. Then we can compare these corrected residuals for these green foreground lineages with those for the black background lineages here. And what we're looking for in our ultimate test statistic is comparing the distributions of foreground relative rates with the background relative rates. To conduct this test and identify genes that may have been under repeated selection related to diet, my trainees and I use the following data sets. My former graduate student, Katie Foley, with the help of several undergrads, including Funbi Ocho, did a literature-based search for the species that we have genome data for, categorizing mammals as either carnivores or piscivores, where the majority of their diet came from vertebrate animals, or herbivores, where the majority of their diet came from vegetation. And this was primarily based on information from Walker's Book of Mammals. And we combined that information with genetic data from the Zoonomia Consortium, the recently developed coding sequence alignments for hundreds of mammals using the Hiller Group's TOGA algorithm. I filtered these alignments to retain high quality protein coding alignments. And we focused our analysis on the subset of the phylogeny for which there was variation in these extreme dietary cases, so specifically Laurasia. And then I estimated the gene-specific trees, keeping the species tree constant using estimated maximum likelihood implemented in R. Once we ran the RER Converge program to identify genes with different distributions of evolutionary rates in carnivores and herbivores, we then corrected our results. The resulting data set represented roughly 15,000 alignments of protein coding genes that had data available for a sufficient sample size of both carnivorous and herbivorous lineages within our data set. Once we ran the RER Converge software to identify genes with differences in relative rates between carnivores and herbivores, we then corrected for the propensity of relative rates to correlate across clades of the phylogeny using a strategy called permutations. To illustrate this, if you look at the real distribution of phenotypes on the tree, we can simulate matched trees that are expected to have no relationship to our phenotype, where the phenotype evolves randomly with respect to the tree. And we can compare our genome-wide results for real diet phenotype with these matched null phenotypes to get results. One of our top results, once we correct for this correlation across the phylogeny, is a gene called cis-D1, which is a mitochondrial protein that is involved in a mitochondrial protein that associates with iron-sulfur clusters. Once we correct for these correlations across the phylogeny in this manner, one of our top results is the protein cis-D1, which is a mitochondrial protein that associates with iron-sulfur clusters. As you can see from the gene tree here on the left, many of the internal branches leading to large clades of herbivores or carnivores evolve rapidly in the herbivorous clades and slowly in the carnivorous clades. And here on the right, I'm showing a distribution of all the lineages that are labeled as carnivorous or herbivorous for this gene. What you can see is that our corrections for correlations across the lead to an extremely low p-value for this gene relative to all other genes in our set. As another way of scanning through our results for potential results of biological significance, we also looked at the tissues in which our genes were expressed. So what we did was we took the publicly available GTEx data from humans, and we assigned genes to each of the tissues that were sort of at the top of their transcript per million expression patterns. One interesting result is that genes that are expressed in the brain and the thyroid tend to evolve more rapidly in herbivores than in carnivores. And there are a number of other potentially interesting results related to tissue specificity within our overall patterns of relative rates in these genes. We have several planned subsequent analyses and experiments that we want to follow up on these results. So for one thing, we would be interested in expanding our RER converge analyses to other dietary phenotypes and comparing these results with those for single genes versus gene families, as well as patterns of adaptation within our own species. And as I mentioned, I'm very interested in comparing molecular evolution patterns with those of differential expressions in specific tissues of relevance to diet. We're also planning to follow up experimentally to determine what the results of these herbivore-specific sequence changes might be on the structure and function of this top gene, as well as looking at sets of metabolic pathways that operate at the single cell level, testing the effects of different orthologs of those in the yeast model system. With that, I'd like to thank all members of my lab, especially those who contributed and are continuing to contribute to this project. The Zoonomia Consortium, in particular, Michael Hiller and Bogdan Karolenko for the alignments and Nicole Foley for the phylogenetic tree, and ongoing and continuing collaborators, Irene Kaplow, Maria Chikina, Ali Graham, and Emily Puckett. Thank you so much.